Hi, good morning. This is Rick Jones. I'm the Director of Global Flow Cytometry for NeoGenomics Clinical Services. And today we'll be talking about validation and globalization of flow cytometry assays for use in clinical trials. So let's step into it. Topics to be discussed in this presentation include the validation design to meet the intended use of the results, the requirements for successful multi-site global deployment of flow cytometry assessments, information needed to determine assay complexity and regulatory requirements, critical elements to ensure data consistency with global multi-site trials, and the use of global SOPs and analysis templates to be used for consistent results. Now the goal. The goal is to have consistent global longitudinal data throughout the course of the study. And that's really true whether it's a single site trial or a multi-site, multinational trial. But for the purposes of this presentation, we'll consider it as global, meaning multi-site trials. And the journey itself is really what is involved and how do we do it? How do we get there? So strategic placement of capabilities to support trials globally is really the key to the performance. At NeoGenomics, we have laboratories placed for this reason, not only in flow cytometry, but in other methodologies as well. And having the technology footprint in the sites is only step one. So when assessing how projects may be applied globally, or using this term to mean across multiple sites, there are criteria to evaluate. The capability of the organization or the lab, the expertise of the staff and the leadership, the consistency of the data, and of course, quality. Consistent assay performance is required for single phase and longitudinal trials. Due to the limited stability of peripheral whole blood and bone marrow specimens, it may be necessary to implement flow cytometry testing in multiple testing locations across the globe or across the country. When these instances arrive, we need to have confidence that the data generated will be reproducible and reliable across these different locations. Harmonized methods, both the process method itself and the analysis method, a standardized instrument platform, a robust instrumentation calibration process, and reagent control are all very key aspects. You'll note that a key term in all these is either standardization and/or harmonization. And this is really the number one priority in flow cytometry that leads to good data, is eliminating those variables. And why is that? You can set your instrument for very consistent PMT voltages or outputs. You can use bright reagents for dimming engines, and you can make the data look pretty during analysis. But it doesn't mean that the results will be consistent over time. Commercially available reagents take care of some of the antibody issues that can be encountered. However, the instrument is set up, is wholly dependent on the labs running the assays themselves. So there's a very famous book by Stephen Covey called The 7 Habits of Highly Effective People. Habit number 2 is to begin with the end in mind. And it bears a great deal of relevance in this instance. This is a key statement in life, in business, and in this case, flow cytometry as well. So, in order to begin with the end in mind, here's the goal: consistent laboratory results across labs. Even a simplistic example, such as a basic TB and NK assay, can be affected by the differences in process, but a harmonized method and execution ensures consistent results. In the example provided, 3 NeoGenomics laboratories are compared for consistency: 1 in the US, 1 in Switzerland, and 1 in Singapore. As can be seen with the %CV comparisons, all the values are in high agreement across all the laboratories, and show data consistency with the highest percent CV across all locations at 7%. Now, this works also in much more complicated processes. So this becomes even more crucial with more complex analyses, such as minimal residual disease testing or very complex immunophenotyping. The examples shown here are for a cross-site evaluation of CLL MRD across laboratories in California, Switzerland, and Singapore. Only a few examples of the histograms are shown for brevity, but the final result of each comparison is also shown. And though there are very minor differences in the visual representation, the end results show very consistent data across laboratories. And this be both with sample 1 at or near MRD level and sample 2 with a high level of disease burden. In this representation, the data is displayed comparing 25 sample results of the same specimens across the laboratories, showing a high level of concordance in the data between the sites. It's noted that samples at all levels of disease were chosen for this assessment, be it high positive, negatives, and those that are at or near the minimal residual disease level. So in order to build a process for successful assay development, there are some aspects that should be implemented. First, we must establish the assay requirements themselves. This is achieved through sponsor and provider interaction and communication. Cannot stress this point enough, as it is very key to do it right from the very beginning. You don't want to be midway through the validation only to get very pertinent information later and have to start over or even do a full redesign. This includes determination if the method is needed at multiple sites as well. Knowing this from the beginning minimizes the caveats that could cause snags in the process. This loses the one commodity that you can't ever get back, and that's time. Next, we have to design the assay and the reagents that will be used. And we have to pay close attention to the spillover between the channels that could potentially lead to poor resolution, and plan to place those dim or low expressing antigens in bright fluorochromes for best performance. And then evaluate each reagent on its own for consistency. Then we move to the method validation and the SOP development. We must define what is being validated and ensure a consistent process throughout to ensure consistency further down the line. The next stop is a harmonized analysis template. And this is very important to ensure each sample throughout the course of the study, whether at a single site or globally, is analyzed in the same way with the same outputs. And last, we have to consider the transfer of established method to other sites if needed, and ensure continuity in how the assay will be implemented for clinical testing. In order to ensure that our assay objectives are met, we must consider the validation process as a gateway to the data consistency. This is a very deliberate process with many checks and balances that are not to be taken for granted. Will the assay be a customized assay validation or a transferred assay validation originally developed in another location or by another laboratory? You will see that the steps involved for an initial assay validation and that for an assay transfer validation are very similar and deliberate. And in order to consider and ensure consistency, stops in the process to review and evaluate each step are critical to its implementation. These include the stage of the validation plan itself, as well as the initial review of the experiments to ensure you're on the right track for best performance. If multiple laboratories are to be performing the same testing, sharing of samples for cross-site validation may be needed. While I cannot minimize the complexity of the logistics for these assessments, It is a critical part for ensuring the harmony of the results, especially if these data are to be used for patient treatment decisions. Now, there is a difference in knowing that you have a predictive biomarker and thinking that you might have a predictive biomarker in a particular assay. And really, this must be parsed out with the trial data itself first. Typically, in these cases, an exploratory assessment is sufficient in early stage trials. As the data evaluation are reviewed with the intent of further development, the regulatory impact may need to be examined. Does the data from the assay define a clinical response or have implications on enrollment? If the answer is yes, further development and validation will be needed to fulfill the regulatory requirements. If the assay is determined to potentially have an ability to aid patient treatment decisions, then this additional validation will be needed to ensure accuracy, linearity, and limit of detection, to name a few, before it fully becomes a lab-developed test or something to be used for patient treatment. Now, a quick break. This is a tale about a very simple 4-color flow cytometry assay, but one with the intent to be developed into a companion diagnostic. There are lots of representations of successful assays with scores of data to support their claims, and there just are less discussions on how to learn from failures or conflicting information along that path of development. So a few years back, when I was with a different provider, I was involved with development of a rather simplistic assay with the potential to be used as a companion diagnostic using flow cytometry. Initial development was performed by the client and a collaborator, and it was requested that our lab continue with the assay as designed, indicating a predictive readout in the treatment of patients with stage 4 non-small cell lung cancer. According to the FDA, however, the assay wasn't robust enough nor sensitive enough in its current design. However, they did provide very little guidance as to what would aid in that path of development. Long story short, the FDA continued to request more and more intended use specimens to evaluate, and the assay was determined to not be robust or have been developed correctly. In the end, 4 years later, and 650 some odd patient specimens, and we had no assay, all due to stubbornness by both the sponsor and the laboratory to stay the course. This is a real life experience that if things aren't done correctly from the beginning, things can go wrong. So in order to establish standardized processes and methods for each element of assay performance, it all starts with standardizing and/or harmonizing the aspects that can lead to variability. And in order to ensure best results, consistency is the primary consideration, and that means eliminating these variables. We need to take all of these inputs, test each one individually, and then pour them all together before implementing. These include instrument setup and the QC of the instrument, the reagent and the QC of the reagents themselves, the method itself and the SOP that drives that method, and the analysis processes. In the end, all of the aspects must come together, whether at a single lab or multiple labs, to ensure data consistency throughout the course of the study. Now, here's a real-world example. This publication was part of a series of papers with the same goal: to harmonize flow cytometry in clinical trials. There were multiple aspects that were harmonized across laboratories, including providing predetermined lyophilized antibody cocktails to each of the participating labs, as well as providing each lab with aliquots of cryopreserved PBMCs from the same patients using the same logistics. A list of specific parameters and cell subsets was provided to the labs to ensure that all groups would be evaluating the same combinations of markers and cell subsets. There were, however, several other items that were left to chance with the assumption that they would have a lesser impact on the results. These differences were the actual staining methods having been similar, but no definitive direction on how each step should be performed. These included potential differences in incubation times and temperatures, lysis reagents to be used, wash buffers, etc. Different instrumentation types and configurations were all capable of the analysis of the fluorochrome combinations that were provided, but these were not consistent amongst the different labs. And there was different analysis software and different analysis templates implemented individually at each site, which then led to reporting discrepancies. And the take-home message from all of this is that all laboratories involved must be on the same page and agree to all aspects of the analysis. There are software packages available from manufacturers to set up the instrumentation. However, they do not necessarily address harmonization across different cytometers and different sites. There will always be differences in performance between different cytometer types and within the same cytometer type if the procedure is different in setup or configuration. And simply using the same reagents along with the same instrument type with the same configuration within and across locations by itself is still not enough to ensure standardized results. So the first aspect we want to address is the instrumentation itself. There are things with the instrumentation that you can control between instruments of the same type when the configuration is standardized. There are also variables that will be different between instruments, regardless of these checks and balances. The key is to have a calibration procedure that aligns the output of multiple of the same type of instrument to produce the same output to be used consistently from day to day. It is possible to have different instrument platforms and have them perform consistently, but there will definitely be difficulty to align these outputs. It's not usually recommended to use different instrument platforms across different sites for this very reason. The next aspect is reagent qualification. It's an extremely important factor in the longitudinal data consistency. This cannot be overstated for clinical trials in which patients are typically tested multiple times over the course of the study. The reagent should be evaluated for performance consistency as a single antibody as well as in antibody combinations. To ensure consistency across locations, a common source for the reagent should be secured to enable distribution of the same reagents to multiple locations. This can be achieved through either a centralized location or which can then be used to confirm the validity and performance of the reagents, or via a centralized distributor if a predefined criteria for reagent use are defined by the transferring laboratory. Lot-to-lot evaluation and comparison is key to the longitudinal performance of each individual component as well as the full put together panel elements. When choosing the fluorochrome combinations itself, it's really important to really determine potential issues with different combinations like the density, the emission, and even the spillover capability of properties. Brighter fluorochromes should be used for dim antigens and for quantitative results, whereas other fluorochromes can be used for basic phenotyping markers and other markers not requiring the same level of sensitivity. We also must be very well and keenly aware of the spillover properties of each fluorochrome to be used in combination and optimize the instrumentation to minimize the over or under compensation of these key markers. This is really especially important when observing dim antigens. Now, it's widely known that some dyes are more stable than others. And here's a brief example previously presented some years ago of what can happen when tandem dyes start to break down due to light exposure and or temperature fluctuation. In example A, this is an example of some intracellular cytokine data showing some false positive results when dyes start to degrade with PE-Cy7 and APC-Cy7. It's important to note that these examples are unstimulated cells that should show little to no cytokine positive events. However, there are positive events being detected in both. In line B, the removal of the APC-Cy7 reagent that was contributing to the false positives in the APC channel now disappeared. However, the interferon gamma APC reagent is still being stained. This means that if the results were real in example A, the interferon gamma positive results would still be detected in example B. The same is true with PE and PE-Cy7. However, both are present in both aspects. Another example generated in our laboratory is shown here. In this example, a common commercially available 6-color immunophenotyping cocktail for the detection of TB and NK subsets is shown. The material is shown as old lot, was a lot in use that was starting to show some atypical results that were not always apparent with the standardized gating applied as used with the IBD protocol for the material. In this example, the PE-Cy7 component was showing signs of degradation when compared to a new lot in parallel testing with the same patient sample. When this occurs, cells bound with the degrading PE-Cy7 material may show as positive events in the PE channel. And when plotting this PE-Cy7 versus PE, this is really readily observed, but it may not have been initially detected in the original analysis template, which could have led to the assumption of a high level of NKT cells rather than the degradation of the cocktail itself and the false positive events. One thing that can help identify this is the use of control cells. While that's not a primary topic of conversation, it is a very useful tool in determining consistency of results throughout the course of the study. Next, we come to the actual SOP itself, and understanding the validation is crucial for implementation of highly complex assays and how they be used. Does the assay generate reliable and reproducible results? Does the assay provide all qualitative or quantitative results? Does the assay address intended use population? Does the assay fit the intended purpose, whether that be exploratory or for patient treatment decisions? And are all of the associated regulatory concerns addressed? In the end, a single process or SOP should be created and implemented and shared across all testing sites, and training documentation being very key to support consistent assay performance. Proficiency testing is another way to ensure performance between sites and should be monitored regularly. So we've controlled the sample preparation with the reagents and the procedures themselves. And then we've established the instrument qualification and calibration procedures to ensure consistent acquisition and the cytometer. And now we must move into the development of an analysis template that can be used consistently and effectively whenever testing is performed to ensure consistent results. So even if all other aspects of assay performance are standardized, the data analysis itself can introduce the single largest error of inconsistency in flow cytometry results. What populations will be measured and how will they be reported? This includes the gating hierarchy and the parent population, and whether intensity measurements will be addressed. Every parameter should be confirmed to ensure that all data will be reported is consistent and accurate. How can we ensure consistency in gating across locations? For one, the use of a standardized analysis software and a shared analysis template is the greatest line of defense for discrepancies. But this can't stand alone by itself. Analyst to analyst variability should also be reviewed regularly to ensure consistency as well. This can also be mitigated by using a centralized analysis approach by analyzing all global study data by the same analyst in a single location if needed. Now, for consistent assay performance, it requires adherence to these global processes and SOPs. All sites must use the same SOPs. All sites must use the equivalent reagents that have been QC'd in the same manner. All sites have to use consistent instrumentation and set them up consistently across locations. And all sites must analyze and report the results using the same analysis technique. These points in blue are also required for predictive biomarker analysis, but would also strengthen exploratory or RUO assessments as well, many of these being regulatory requirements. And now we return back to the first example from the beginning of the presentation. Again, bringing in to begin with the end in mind. The data displayed are the same 6-color TB and NK assay as shown previously. As displayed in the course of the presentation, the results can be affected by differences in performance, but a harmonized method and execution ensures consistent results. These data would not have been achieved the intended results without the processes in place to ensure consistency across the labs, be it labs separated around the globe in this case, or even 2 labs across the street from one another. So in conclusion, we need a clear understanding of assay requirements and the intended use of the data because it is paramount to the successful assay validation deployment. Consistent global assay performance requires harmonized processes for instrumentation, reagents, SOP, and data analysis. Applying these harmonization procedures allows for very consistent data between instruments and across multiple locations. And consistent data between instruments and across multiple laboratory sites is really dependent on these harmonized assay-specific procedures. I do want to thank you for your time today. I do have a few acknowledgements I'd like to say on behalf of the team. The Brian Moe in California, Ben Franke in Switzerland, Phil Davis in California, Michael Chinyo Kang in Singapore, and Richard Hughes in the marketing department in the UK, as well as a consultant for us, Dr. Ken Penline from Red Consulting. And of course, the entire Oxford Global Conferences team for having this platform available and bringing this all together to learn and discuss. Please join us later for the Q&A session, which I believe is set to follow this presentation. And I do thank you for your time. If you need any further information or questions, there's a way to reach out to me on this link provided below. And thank you for your time today.